Assalamu alaikum and a very good evening to everyone. On behalf of Vitanov International Alliance for Sciences, I warmly welcome all our respected speaker, distinguished guests, and participants from around the world for Class 3 of the Advancements in Molecular Biology course 2026. We are delighted to have you with us once again as we continue this exciting learning journey in the modern molecular biology. Today's session is titled Codon Usage Bias Analysis, a fascinating topic that helps us understand how organisms preferentially use synonymous codons, and how this knowledge contributes to gene expression studies, evolutionary biology, synthetic biology, and biotechnology. It is my pleasure to introduce today's estimated instructor, Professor Dr. Iftekhar Ahmad Baloch from Balochistan Academy for College Teachers, Pakistan. We are truly honored to have such an experienced academic and educator sharing his knowledge and expertise with us. And without taking any more time, I would like to invite our respected instructor, Professor Dr. Iftekhar Ahmad Baloch, to begin today's session. Sir, the virtual stage is yours. You can now start your session.
Thank you very much. So I hope all the participants present here are having some background in molecular biology and bioinformatics. So this lecture is not a very advanced one. I have designed it for the beginners. And I hold a PhD degree in botany, so I used some bioinformatic tools. And this lecture is introducing one of the bioinformatic tools used for codon usage bias analysis. So I want this session to be interactive. This should not be monologue, but dialogue. So if the participants are hearing, so I may ask a question to them that how many of them are familiar with this term codon usage bias analysis or CUB analysis? Anyone here? Hello? Am I audible?
Participants are requested to
Hello?
write in the comment section their answer.
Okay. So their mics are muted, I think. So no matter. I just wanted to have this session to be interactive. So let's take start from the very basics. Codon, everyone is familiar with the term codon, that it is a sequence of three nucleotides in the messenger RNA or mRNA that specifies a particular amino acid. So besides these amino acid sequences, we also have three stop codons, and everyone is familiar with those stop codons, 61 codons and three stop codons. So these 61 codons code for amino acids, and on this side I've put a codon wheel. So this is not a horoscope wheel. This is a codon wheel which tells about how many codons are used for a particular amino acid. So this is just the basic. And here comes the codon usage bias. As we know that there are 61 codons available in eukaryotic organisms for coding 20 or 21 amino acids. But these codons are not used equally. So codon usage bias refers to non-random presence of the synonymous codons. It means that different codons that encode the same amino acid in the genomes of the different organisms. Here is an example that multiple codons can encode same amino acid. I hope codon degeneracy, codon redundancy, these are basics, so everyone is familiar with them. So organisms often use some synonymous codons more frequently than others. For example, the amino acid leucine is encoded by 6 codons: UUA, UUG, CUU, CUC, CUA, and CUG. For the last 41 CUU, CUC, CUA, it is often written as CUN. N means that any of the four letters CU, either it is CUA, CUG, CUC, or CUG. So there are six amino acids, six codons available to code amino acid leucine. But interesting thing is that an organism may preferentially use CUG more than the other leucine codes. There are six codes, but some organisms prefer to use CUG instead of rest of the available codons. So this is a sort of bias. This is a sort of preference why an organism uses this particular codon instead of using the other one. For example, if there are four codons available for a specific amino acid, so an organism should use 25% of the each codon, but it does not happen practically. But organisms prefer to use a particular codon. So this preference is known as codon usage bias. So there is a bias in the usage of these codons. So why does this codon usage bias occur? Codon usage patterns are influenced by tRNA abundance. As we know that tRNA, they bring amino acids, and to the messenger RNA they harbor there, but there are not so much transfer RNAs as there are 61 codons. So this number is different, and sometimes this is done by natural selection for efficient and accurate translation. Genome composition, some genomes may possess GC regions, and some are rich in AT content, mutation pressure, gene expression level, and evolutionary history. So there are a lot of applications of this codon usage bias. Why are scientists interested in analyzing the codon usage bias of different organisms? So codon usage bias analysis helps in understanding genome evolution, studying gene expression patterns, and optimizing gene for recombinant protein production. I will focus on this third point, and rest of the things are also there, but we will focus this third one. The fourth one is vaccine and biotechnology research, and fifth one is comparative genomics and phylogenetic studies. So how does CUB, I will use the term CUB instead of codon usage bias, how does CUB analysis helps in understanding genome evolution? So different organisms have distinct codon usage patterns. By comparing CUB profiles among species, researchers can identify evolutionary similarities and differences between organisms, and genetic relatedness among species, possible evolutionary divergence events. For example, closely related bacteria species often show similar GC content and codon preference, while distantly related species show different patterns. Another application of CUB is that it helps in studying gene expression pattern. So some genes which are highly expressed genes often show strong codon bias because natural selection favors codons that allow rapid and accurate protein production. And similarly, frequently used codons usually correspond to abundant tRNAs. Genes using these preferred codons can be translated more efficiently. Example, in bacteria, genes encode ribosomal proteins often show a strong codon usage bias. They need to be produced in large amounts. And this CUB analysis also helps in optimizing genes for recombinant DNA protein production. So, recombinant protein production involves expressing a gene of interest in a host organism, such as E. coli, yeast, insect cells, or mammalian cells, to produce a desired protein. A major challenge is that codon preference of the foreign gene may not match the codon preference of the expression host. CUB analysis helps identifying and modifying the differences to improve protein production. And this CUB analysis also helps in vaccine and biotechnology research. Codon usage bias analysis is an important tool in vaccine development and biotechnology because it helps optimize gene sequence for efficient protein expression, which improves vaccine efficacy and understand pathogen evolution. So, another application of the CUB analysis is that it serves as a genomic signature that complements sequence based analysis, providing insights into genome evolution, species divergence, host adaptation, and horizontal gene transfer. Although it is not a replacement for sequence based phylogenetics, but it is a valuable complementary approach in comparative genomics. So, as I said, that we will focus the third point of the codon optimization and production of protein, expression of protein in another organism. So this is a diagram showing that if you want to get a human gene to be translated in E. coli, so before doing this, codon optimization is must, because both these organisms use separate types of codons for particular amino acids. So some of the organisms, they use codons which are very rare, and these rare codons can cause premature termination of the synthesized protein or disincorporation of the amino acid. As you know that a single difference of a nucleotide can matter a lot, as it changes the reading frame of that particular mRNA. So this reading frame mutation and reading frame shift can cause a lot of problems, and it affects the translational efficiency also. So here is a step-by-step explanation of a gene to be incorporated in a E. coli, and we want to get it expressed in E. coli. So this is a practical example. So if you are having access of net now, or later, all the facilities, if these are available, there's a resource called CDI. You have to download it, and then you have to sign up for it. Then you can do this practically, and you can understand the concept and its application. So the first step is retrieve the gene sequence from NCBI. I hope everyone is familiar with NCBI, National Center for Biotechnology Information, where it's a very big resource, and I have used only some of its tools during my work. So one of the features it provides that you can fetch the sequence of different genes from this NCBI. So in this example, I am interested in fetching the Homo sapiens, insulin gene from Homo sapiens. So if I type the gene ID, that's 3630, in its search bar, it can fetch the amino acid sequence, and it can also fetch the nucleotide sequence. So it fetches both the sequences. And if I'm interested to get a human insulin gene into a E. coli, so what should I do, and how to do the codon optimization before that practical work. So for this codon optimization, I should go to the NCBI. I should fetch the sequence of the insulin gene. So in the next step, uh, there is a reference mRNA. So reference mRNA number is also here, given here, NM 000207, and protein ID is also there, given there. But what should I do? In step 2, I will open the reference mRNA instead of the genomic sequence. That transcriptomic sequence, reference mRNA is preferred. Why? We will discuss in next slide. So click on the RefSeq transcript and open direct, or directly open from the insulin gene page. So you can have its FASTA format also there in the GenBank, and in GenBank you can have its coding features, coding sequences, CDs feature to highlight the codon region. Here is the example. When I fetch this the sequence of this gene, so I will be having this nucleotide sequence. But if I am interested to know the only sequence which is protein coding, because a sequence may contain upstream and downstream regions, which are 3' untranslated region and 5' untranslated region. So I am interested only in the sequence which is coding for the protein. So therefore, we can highlight those nucleotide sequences which are coding for the protein. The third step is that copy this sequence, the coding sequence, for the codon optimization, and use coding sequence rather than entire mRNA, because the entire mRNA contains 5' and 3' untranslated regions, which are known as UTRs, that are not translated into proteins. So this is the graphical representation or pictorial. At 5 prime, everyone is familiar that at 5 prime of the mRNA there is capping, then there's a untranslated region, and then there is a coding sequence or CDs, and again there is a 3 prime untranslated region, and poly A tail is there on the 3 prime end. So instead of fetching the entire sequence, we are interested in fetching the coding sequence only. So in the step 4, we download the sequence, and we can save it, we can download it, and or we can simply copy and paste it to the next resource, to the next bioinformatic tool which we are using. So after doing this, the next one is uh, itdna.com. So that is integrated DNA technologies. They have prepared a software which helps us in understanding or doing our codon optimization before practical, before using the wet lab, and before synthesis of that particular sequence for a vector. So step 5 is open the IDT file, cursor or arrow on it. So you can do it by searching the website. It has got a search option there. So IDT codon optimization tool facilitates you to do your codon optimization. So step 6 is select expression host. The sequence you have fetched from human insulin sequence, the sequence you have fetched from a different organism, and the organism which you are interested to get it expressed in. So these two organisms use different codon tables. So when you select the organism, so select the expression host. So there's a drop down menu which facilitates you to select the organism. E. coli is given in that drop down menu. So the software loads codon usage bias table specific to E. coli. And then when you click on the optimization, there is a button given. I couldn't get that screenshot. So you can click on optimization. So what does this software do? It not only optimizes the codon according to the host organism, but it also evaluates the codon adaptation index, GC content of your sequence, rare codons, codon frequency restriction, enzyme sites where you can cut those sites, repetitive sequences, mRNA secondary structure. Remember that mRNA also has a secondary structure. So this codon usage can also, codon difference can also disrupt or entangle that particular folding of that particular RNA. And it also indicates the unwanted motifs. So in the next step 8, the results are with you. I want you to do it practically later. So it tells you that original sequence which you provided, approximately 0.55, its codon adaptation index is this, and after optimization it is indicating the new sequence. It also telling the GC content of that your sequence, and after optimization, which is balanced for E. coli, it tells that rare codons are also present, and it reduces after optimization, it reduces the rare codons. So protein sequence remains same, which is our purpose, which is desired by us, that protein sequence should not get changed. So only synonymous codons change, the amino acid sequence remains identical. So the next step, step 9 is there. So you have to export the optimized sequence. The optimized DNA sequence can be downloaded in the format, in the FASTA format, and used for gene synthesis. As you know that commercially gene synthesis is being there. So you can do go for cloning, and you can have recombination protein expression. And when you are going to clone this sequence into an expression vector, so these are names of vectors given here, and transformation into E. coli. Usually this is BL21 and D3. As you know that when you want to get expressed a sequence into another organism, so you put it in a vector and then you insert it in that organism. So the next one is protein expression. So when you grow the transformed bacteria and induce expression commonly with this vector, the optimized insulin genes, INS stands for insulin, so it is translated more efficiently, producing higher level of recombinant insulin. So I hope that with this single example, this is a vast field. There are a lot of applications, but I've selected this example to make you familiar with this its application, one of its applications. For the beginners, this is, I think, very interesting, because it is more focused. Instead of focusing a lot of applications, I just focus this one. So another thing which I studied before I did PhD in botany in CAP, because I was working on the microRNAs. So I hope every one of you is familiar with the microRNA. MicroRNAs are a small piece of RNA, small RNAs as indicated by their name. So these microRNAs regulate protein expression along with the RISC RNA-induced silencing complex. They just harbor on the messenger RNA, and they then silent the expression of a particular protein. So here I've put an example, an interesting example, that we sometimes just think that sequence on the messenger RNA is just a sequence to code protein, but it is also a site for harboring of the microRNAs, and sometimes its own secondary structure is affected by the difference of the codons. So here is an example. If you, I am going to magnify it. Now you can see there is a fabulosa mRNA. Here is its cap, and here is its polytail. So there are a lot of amino acids: glycine, methionine, lysine, proline, glycine, proline, and asparagine, etc. So if there is a mutation on the second nucleotide, GCU, if it becomes, instead of GCU, it becomes GAU, what happens then? For example, this one is glycine, and glycine is being coded by GGU, and again the aspirin is there. So aspirin is coded by GAU. So this mutation, or this difference of the single nucleotide, changes glycine GGU into GAU. So it becomes another amino acid. So this affects the growth of the particular plant, that is Arabidopsis thaliana. So, but in another study, scientists who are working on the microRNA, they found an interesting phenomenon. They just experienced that if this GGU, which is coding for the glycine, if it becomes GAU, then it codes asparagine. When it codes asparagine instead of glycine, so the growth of the plant becomes stunted and it becomes deformed. Many deformities are observed in this plant. Now, what happens? Now, see, if this U is replaced with the A, say if it becomes GGA, if this mutation is on the third nucleotide, it still codes glycine. I mean to say that glycine is coded by GGU, and the same glycine is coded by GGA. So these are synonymous codons. But instead of having the same codon and the different codon but coding the same amino acid, so it means the protein should be same here. But astonishingly, scientists, they found that again this growth structure and the deformity is observed in that plant, Arabidopsis thaliana. So why does it happen? So it happens because a microRNA, when it harbors a messenger RNA, so it cannot bond there properly. So I hope you understand that besides just coding amino acid, the mRNA sequence affects a lot of things. One of the examples I just put in the slides related to the microRNAs. Now you can see that how important this thing is, and codon usage means a lot, instead of just coding the sequence. As you know that micro mRNAs also, they form a secondary structure. Same is the case with the microRNA, but this secondary structure is also affected by usage of the codons. When a codon is used with its synonymous form, so it also affects the secondary structure of microRNA, a messenger RNA. It affects protein expression of a messenger RNA, as I quoted in the example. When a microRNA harbors, even then the amino acids are same amino acids are being coded, but with the different nucleotide sequence. So the difference of this nucleotide sequence also affects the growth of a plant. So this is just a short presentation. Thank you very much. If there are any questions, so forum is open. I welcome you all. Thank you very much. Done with it. Hello.
Yes, we are listening you. Uh, Thank you so much, Dr. Professor Iftekhar Ahmad Baloch, for the wonderful presentation. And now, I just want to say to everyone, if anyone have any question related to the presentation, you can write in the chat box. Our moderator will call your question. Yeah, thank you.
First of all, thank you so much, sir, for your organized and board explanation, as well as those slides were very effective for the students to know about the codon who are having the basic knowledge. So right now, as we are on the Q&A session, so I would request participants to ask questions in the chat box. We will wait for a few moments for the questions. Okay, sure. Someone asked that if the slide can be shared in the WhatsApp group of our interns. So, can we share your slides?
Yes, you can.
Okay,
And if
thank you.
there's a WhatsApp group for the participants, they can ask also questions there.
Okay, so yes, the slides will be shared after the meet, after this session.
Okay. So, isn't there any question? And are there graduate level students, undergrads, or MPhil PhD students? So,
Yes, there's a diverse students like from undergrad to PhD, and there's no question, but everyone is praising your presentation and the slides, the way you delivered the insightful informations. So they are, yeah, they are not having any doubt due
so I
to
would be
this.
glad if there are questions.
Yeah, we will wait for 2 to 3 minutes for the questions so that we can clear any doubt the participants are having. Okay, we have a question. That is, what are the applications of codon usage bias in research?
Yes, I think this little portion, it was partially covered in my lecture, that codon usage bias analysis has a lot of applications. So every researcher can do it according to his or her needs. So in one of the slides, I discussed this thing, that if you are interested in understanding the genome evolution of a particular organism, so codon usage bias is there now. As you see, that traditional taxonomy is now nowadays being discouraged, and instead of traditional things, they scientists are interested in understanding the things on the molecular level. Besides this, one of the examples which I discussed in my slides, gene expression patterns, that how many codons are being used for a particular sequence in different organisms. The third one was gene optimization, which I discussed in detail, that if you are interested to get human insulin gene expressed in a E. coli. So before doing this, you have to go for the codon optimization. And if I talk of my area, microRNA research, so I put also an example of the microRNA research, that this codon usage difference also affects a lot of things. Say, for example, the growth pattern in that particular uh, plant ATH, I think it was Arabidopsis thaliana. So a lot of applications, maybe besides this, there are also a lot of applications, vaccine biotechnology research, and phylogenetics studies. So especially optimization is done for different optimization tables. As you know that prokaryotes use some different codons as compared to the eukaryote. So these are some of the applications. Five have been discussed here, one in very detail, and maybe there are more applications also.
Okay,
What
sir. We
software
have
are,
another question.
I'm seeing I can see the questions. What
Okay, then
software
you can address.
online tools are most reliable for CUB analysis? I think all of them are reliable. NCBI is a big reliable resource. NCBI does not offer directly the codon optimization. Maybe it is offering now because this is very rapidly evolving field. I was just astonished when I visited NCBI when I was doing some work on CUB analysis of the salt resistant genes and normal genes. One of my students, she was doing her work, so the interface was very much different. So at NCBI there is a resource available which is reading frame, open reading frame, ORF. So when we were working, so it provided only 6 ORFs, open reading frames for a gene. So now today, before when I was preparing this lecture, so I just visited, it is providing more than a dozen of the reading frames. Definitely some change might occur, and the things which you are doing now, if you visit a particular website 1 month later, so the things will be a lot of different things will be there. As far as your question is concerned, that which software is reliable for CUB analysis, so one of the reliable sources, or say uh, which we discussed, is the one I'm coming on that slide, this one. So I don't know why it is going back again. So this integrated DNA technology, ittdna.com, it provides you a tool for codon optimization. It provides you variety of organisms. It provides you the codon tables also. So you can use it, and it's also free. These resources are often they are free.
Thank you for addressing the question. We have another question,
Okay.
so can you address it as well, sir?
Well, this is an interesting question. What is the effect of change in rare codon frequency on 3D structure of a gene? Do all the rare codons may of many amino acids is recorded, or what? I can't see this portion. Uh, so
Okay,
that's the
sir,
question.
let me read it for you then.
Mm.
It is what is the effect of change in rare codon frequency on 3D structures of gene? Do all the rare codons of many amino acids is recommended?
Okay, very interesting question. Now, first of all, if you are asking about the mRNA, so mRNA folds and it gets a secondary structure. So it does not go for the tertiary or quaternary structure like protein, but yes, it does affect. It does affect the secondary structure and 3D structure of a protein. I'm trying to fetch that paper where we compared protein 3D structures of different genes. If I'm able to fetch it, or if later, if I'm added in that group, I will share it in that WhatsApp group also. So let me search.
Thank you so much for addressing the question as well. I think that we are not having any further questions. So Thank you very much, Professor Dr. Iftekhar Ahmad Baloch, for delivering such an insightful and engaging session on Codon Usage Bias analysis. Your valuable knowledge and practical explanation have greatly enhanced our understanding of this important topic in molecular biology. We also extend our sincere gratitude to all our participants from different countries of our active of your and as well as ours active participation and enthusiasm throughout today's session. As a reminder, attendance link has been given in the comment section. So fill the form for to mark your attendance. And we look forward to seeing you in all our next session as well for the Advancement in Molecular Biology course 2026. Until then, keep learning, keep exploring, and keep advancing in science. Thank you everyone for joining today's class, and thank you our speaker as well for delivering such an amazing presentation in this class. So Allah Hafiz, everyone. Have a wonderful night.
Okay, thank you very much. If I'm added in that WhatsApp group, I want to share a paper later, which addresses the question from that Sadaf, Miss Sadaf was there. So here in that paper, they have compared the protein structures also after that
Okay,
Codon usage bias. So
sure, sure, sure.
done for now, I think. Mr. Murtaza there?
Oh, we have a question left. Would you like to answer it?
Okay, does the amino acid sequence alone, uh, uh, does the amino acid, it's not visible again.
Kind of check
Let,
now.
let me. Does the amino acid sequence alone cannot determine the protein conformations such that the triple codon has to be taken into account? And that can be one of the reasons why we go for omics tech. I think it is complete for this. Jibrin Ashir, I think. Am I right? Is Jibrin there? Is the mic on?
Yes, yes, yes. You can continue, sir. You can answer the question.
So, can I talk to Jibrin?
It's not possible to talk directly. We can share your contact
Okay,
so
okay,
that the
okay,
participants can contact to you for the question and answer.
okay. His or her question is that does the amino acid sequence alone cannot determine the protein conformation such that the triple codon has to be taken into account, and that can that be one of the reasons why we go for the question is not clear to me. What does he or she want to say? The amino acid sequence, if the amino acid is being coded by a different codon, so definitely it does not affect the conformation, I think. And if the amino acid is changed, then there are a lot of charges on different amino acids, a lot of conformations, so it may change. But as far as your question is concerned, does that amino acid sequence alone cannot determine the protein conformation? So I would be glad if you clear your question so that I may able to reply.
Yes, and of course, your sites will be shared in the group as well as your contact information so that the participants can reach out to you for their confusions and questions. So for now, it
Okay.
is, I think, it is a time to end our session. Once again, thank you everyone for joining the session. Thank you, our estimated speaker as well. We
Thank you
look
very much.
forward to, yeah, sir. Would you like to say something as a closing remark?
Thank you. Done. Okay.
Okay, most welcome. Thank you everyone for joining us today. Allah Hafiz and have a wonderful evening.